Awesome. Yeah, Isaac.
Yeah, well, I not the most recent, but a recent data set that I worked with that had a couple of really interesting properties that I think are important for people to think about was a cannabis data set. Some collaborators generated. It was 5 different plates of data that had been sequenced at 5 different facilities. It really was kind of like a kind of perfect storm of bad luck. And so we had these 5 different plates of data, and we wanted to look at, we wanted to combine these plates, and there's a couple of different problems. So one problem, first of all, that I realized early on was that there was a massive amount of contamination from mitochondrial and chloroplast, and so one of the things that iPirate allows you to do is filter out, like use a reference to filter out data. So I downloaded the chloroplast and mitochondrial reference genomes from the internet and filtered out all of the organelle hits. And so that was one kind of like trick that improved the quality of the data. The other kind of issue that we had with this data set that was really nasty is that because they were, the libraries were prepared in different ways and they were sequenced at different facilities, and just looking at this kind of is, I guess, what Alicia was kind of mentioning too, in the same kind of direction. If we looked at the locus in the final data set, if we looked at the locus sharing across all samples, there was almost no shared loci across the different plates. So all the shared loci were within any given individual plate, regardless of the relatedness of the samples among the plates. And so there's this really kind of nasty batch effect, which if you're not aware of it, you would plug this data into the PCA or into some kind of like population genetic analysis, and you get 5 really good clusters popping up, but it's only clustering because of the missing data and because of the lack of locus sharing across the plates. And so this is something, this is just kind of like something that people who are doing, assemblies that are combining runs across different like libraries or across different sequencing experiments need to be really super careful about.
Thanks. Yeah, that's a great point. Jon Puritz was actually the one that I saw a poster of his that you have to split up your samples from different populations across runs, or you could get that exact result. Yeah, Jon.
Yeah, I guess the most recent data set I've worked with was actually with what we thought were 2 different coral species. And we were trying to do some different applications, like adding in some epigenetic inference along with RAD inference. But the assembly is essentially the same, and we ended up finding that not 2 species, but more like 8 species were in our data set. And then so kind of like doing initial assembly optimization and then parsing out into 8 different individual assemblies, trying to handle some of that cryptic variation. So that was one challenge. But I'm glad that Isaac and Alicia both brought up really important things that I think we definitely should get to talk about. But batch effects and its sample variation and coverage, I think, are really huge things that everyone needs to think about. And it's not anywhere that you find, I think, in a lot of the literature out there that you might start to think about. So I just wanted to echo both of those are really important considerations. And the batch effects, if you don't have a high signal system, can really be surprisingly strong in your data set. So.
Yeah, great. Yeah, one of the topics I wanted to talk about was filtering, either as you're doing your assembly and clustering or afterwards. And so maybe we can actually, since you all sort of brought up things that are related to how you filter your data, either iteratively as you're passing samples through or at the end. So yeah, for any of our panelists, what are some of your personal guidelines you use to, as you're getting data working through the pipeline, check to make sure that you don't have a batch effect, you aren't losing a lot of loci because of uneven coverage. You know, what are some of the things that you check for? Yeah, you can just raise your hand. Yeah, John.
So I'll kind of take one step back, and I think for me, a lot of this comes down to some good pre-planning before you start your libraries. If you can, I mean, of course, sometimes you run into a data set that has bridged multiple labs and collaborative groups, and you have to deal with this a little bit later. But the more you can kind of think about this at the design phase, you can kind of help eliminate or at least mitigate some of these circumstances. So one of those is randomizing your samples across library preps and lanes of sequencing. If you're going to have multiple lanes of sequencing, that's one great way to help with any potential batch effect. The other thing that I always recommend to people is replicate samples. Like have the same 3 to 5 samples that you sequence in every library prep and every lane of sequencing, because that's the best way to detect whether or not you're having batch effects. Because if something's not genotyping the same way in the same sample, you know something is wrong. And so that's like my kind of pre-filtering before even getting to those batch effects. So I'll turn it over to the rest of the panel to kind of talk about some of those other issues, but that's definitely one of what I mean by that.
Oh, sorry, Isaac, and then Alicia.
I think Alicia was maybe before me, but
okay, yeah,
do
Alicia.
go ahead,
Then
I raised my real hand instead of
no,
yeah,
that's okay. Yeah.
All right, thank you, Isaac. Yeah, I just want to echo the importance of randomizing samples. It's horrible. It's you spend so much more time doing it, but it's worth it. Everyone hates doing it because of the extra time involved and the extra careful of not mixing up stuff. But I think it's worth it. And I will say that if lab work is being done by someone with not a lot of experience or that is learning, or if the samples are quite precious because they were difficult to get from a particular population, I will also randomize them, since DNA extraction, or more than randomizing, just don't extract all samples from a single locality or any single treatment or something that could be generating a batch effect together. Just mix them so that you avoid batch effects at the quality level of the DNA. This is something that we have seen too. And it's just more time in the lab, which is already taking time, but I think it's worth it. And something we also do, I love PCAs because they are great at just telling you if there is something terribly wrong. You are gonna see it on a PCA. So it's quite obvious, but just plot your data as soon as you have the first plottable data, even if it's not the final filtering, just plot it and color it by all the possibilities you can imagine, by locality, by species, and by batch, by sequencing lane, and by who did the DNA extraction, all these things. And I do that. That's the first thing we do, like as soon as possible. And if there is something wrong, you will see it there.
Yeah, go ahead, Isaac.
Yeah, well, first of all, I totally agree with Alicia and John about randomization and replicating samples if it's possible. I know that this is like not always possible just because, you know, replicating samples doubles the amount of sequencing you need to do, but it does make a huge difference. I also agree with Alicia that using PCA to just diagnose, like as a kind of like rough diagnosis, is super useful. It's really nice for I can't count the number of times where I've caught bad samples or just kind of weirdness in samples just from a quick PCA, and things pop out and you look at them and it's just a really nice way of, like, you know, rapidly evaluating the data. In terms of like the kind of broader process of how to approach filtering, I think that from, you know, what I like to emphasize is that filtering isn't, you know, something that you kind of do at the end, right? Like you have to kind of like it's really important to evaluate the results of each kind of step of the assembly process. And so I'm going to put this a link in the chat to just some documentation. We run workshops where we kind of teach people how to do iPirate assemblies and analyze RAD-seq data. And this is kind of one of the things that we emphasize in the workshops is like looking at your data is super important. And you know, it's tricky because like I think that you have to when you're evaluating the quality of RAD-seq assembly, you can't approach it like it's just a like massive multi-locus data set, right? Like you can't sit there and look at all your alignments by eye. So you have to develop different kind of heuristics for how to evaluate the assembly quality at each step of the process. And so we can't go into all the details right now, but you can look at this link where we kind of help people have iPirate generates like lots of different statistics for assemblies at each step of the process. And so the RAD-camp documentation helps people evaluate, think about the statistics at each step and how sequence quality, sample quality, or assembly parameters can impact the assembly quality at any given step. So
yeah, awesome. No, that's really really helpful link for both the website and I'm sure for everyone here. So thank you for sharing that. Melanie.
I just wanted to add, going back to the library effects, that if you're able to randomize your samples among libraries, you can use outlier detection to try to deal with some of those library effects. So for example, one of my projects had a species that had very low levels of differentiation. I thought I had 5 clusters because it turned out that it was 5 sequencing lanes that were driving the structure that I saw in the PCA. And so I was able to use BaseScan to do outlier detection and remove some really high FST SNPs, and that collapsed all the clusters together. So that's a nice method, but it does require that you had randomized your samples, because yeah, if all of your samples from one geographic area are in a single lane, you won't be able to tell whether that structure is from geography or from a sequencing effect.
That's a great idea. Yeah, thank you. And so, we've sort of mentioned.
Can I just add on to that?
Yes, yeah.
Fast? So yeah. I think that's a great approach. If you do have randomized samples, outlier approaches will find some of those library effect loci. And the other thing that I recommend to people to do, if you are going through that approach, is to actually visualize some of those alignments of those high effect loci. You may actually even be able to see in the most of the time when you see a wonky alignment, you'll know that something's wrong. And so you can, I always encourage people to, when you're doing some of those outlier detections, to find some of those potential batch effect loci. Take a look at the alignments, and I bet you'll see what is potentially generating that problem that's there. So just wanted to tag that on because it's a great suggestion.